class. Today I'm going to be talking about DNA replication. DNA is a genetic material of all living organisms. DNA is composed of 4 different nucleotides, which are adenine, guanine, thymine, and cytosine, and a sugar phosphate backbone. The purine bases adenine and guanine pair with the pyrimidine bases thymine and cytosine, and this is known as complementary base pairing. Specifically, adenine pairs with thymine and guanine pairs with cytosine. These bases are held together by hydrogen bonds, right in between two bases. The structure of DNA is a double helix, which is composed of the 2 sugar phosphate backbones on the outside, and on the inside, the paired bases. DNA is, luckily, a self-replicating material. The process of replication is composed of unwinding, complementary base pairing, and joining. Because DNA is a double helix, the structure has to unwind for replication to occur. The enzyme DNA helicase unwinds the structure and separates both strands of DNA. And the double helix structure looks like this. And then what happens is it becomes unwound by DNA helicase. And when it becomes unwound, it's kind of like if you were to take a Twizzler and unwind it, which I'm sure if you've ever had one, everybody's done that. And it results in 2 strands instead of just one. And these separated strands are called parent strands, and they serve as a template for the complementary base pairing that takes place in the next step. DNA replication is a semi-conservative process, which means that each DNA molecule is composed of a strand from the previous molecule and a complementary new strand. DNA is not structurally stable when it's unwound, and single-stranded binding proteins have to be used to keep the strand from winding back into the double helix shape. It's kind of like what I drew here. They just kind of, stick to it, and they help it not to wind back up, because it really wants to do that, because the inside bases don't really like to be around water, so the sugar phosphate back strands, they protect them from the water in the cells. Next, complementary base pairing occurs. DNA replication needs a primer in place before it can begin, and DNA primase is an enzyme that places these primers, which are small strands of RNA, on the parent strands of DNA that will be replicated, and then of course match up, and they're complementary paired to the DNA. DNA polymerase recognizes these primers and begins replication at these spots. When adenine is present, thymine is paired, and when guanine is present, you guessed it, cytosine is paired, and vice versa, depending on which is present in the parent strand. These bases are paired to the parent strand, and a chain of nucleotides is created in a new strand. In this way, 2 new DNA molecules are being created simultaneously, and the old strands of DNA are being used again. And I drew this here, where you've got the 2 strands, one's new, one's the parent, and you've got the DNA polymerase helping it being paired. DNA polymerase also proofreads the new strands to make sure no errors have occurred. So it's got some pretty important jobs, because if this doesn't happen, there'll be like 1 in 100,000 mispaired bases, and we have a lot of issues with our genes. So it fixes the errors, and it also converts the RNA strands that have been used as primers to DNA strands. So you've got all DNA and not some RNA and DNA, because that would also be kind of bad. And the last step in DNA replication is joining. In this step, DNA ligase acts as kind of like a glue, and it joins the new and old strands of DNA together. So you don't just have these strands floating around. So you get the double helix shape, and it kind of looks like this. And you've got the DNA ligase, which kind of joins them together. and then you eventually have two new molecules. So the DNA replication results in two new molecules that are composed of a parent strand and a new strand. So it's a pretty easy process, but has